Hello, everybody. I'm Aayush. I'm a PhD candidate at ETH Zurich, and today I'll be talking about an attention-based method that we have been developing to predict chromatin interactions in unseen and rare cell types. We know that the DNA can be divided into two kinds of regions, coding and non-coding. The effects of mutations in the coding regions are well studied. However, we still have very limited understanding of the mutations in the non-coding regions. And so the goal of this study would be to kind of lower the gap between this. To do this, we will look at the 3D structure of the DNA. Specifically, we'll be focusing on enhancer-promoter interactions. Trying to predict enhancer-promoter interaction is a very challenging task, majorly because, as we saw before, that enhancers can regulate the genes that are few megabases away from them, thereby forming large complex loops. And as we also saw before, these are also mediated by the TADs. So the elements within the TADs tend to interact with each other a bit more than the ones outside it, and these influence the enhancer promoter interactions. To do this experimentally, we capture the 3D structure through the Hi-C experimental setup, which we saw in the previous talk. So I'm just going to skip through this. The broad goal of this work would be to predict genomic interactions. So we predict values within this Hi-C matrices such that we can capture long-range interactions. At the same time, we would also want to focus on using minimal epigenomic training data sets to predict this. And we want our method to be generalizable to new cell types. The reason why we want them to be generalizable to new cell types is that if we have a cell type for which we do not have enough data to generate or to conduct this Hi-C experiment, we should still be able to somehow do in silico studies on top of it. So there are several existing works that can accurately predict the Hi-C matrices, and these fall into three different categories. The first category focuses only on the DNA sequence. While these kind of methods can predict the Hi-C interactions very accurately in the cell types they were trained on, these cannot generalize to new cell types. The second kind of methods use multiple epigenomic tracks as inputs, and these includes histone marks and CTCF binding sites, and we might not always have all these kind of epigenomic tracks available as inputs. The third ones focus only on the enhancer and the promoters, so they take information only from the enhancer and the promoter regions, thereby not taking into account anything that happens around them. So effects of TADs will not be captured in these cases, which is not ideal. So to bridge these gaps, we introduce UniversalEPI, which only takes the DNA sequence and chromatin accessibility as inputs and predicts Hi-C interactions between regulatory elements across for new cell types as well. UniversalEPI in its essence is a two-staged method where at stage one, we learn the features of ubiquitous transcription factors from DNA sequence and chromatin accessibility. Using these learned features, we then move to stage two to predict the cell type specific Hi-C interactions. Now let's dive deeper into stage one. So in stage one, we predict the binding affinity of various transcription factors. We focus mostly on CDCF, YY1, and SP1. The reason why we focus only on these three is twofold. So one, they behave very similarly across cell types, and therefore we ensure that we do not learn anything that's specific to any given cell type. And second, they all play a role in genome folding. So we know CTCFs are endpoints of TAD boundaries, endpoints of TADs. YY1 is usually present at enhancer promoter interactions, and SP1 are present in GC-rich regions, which can also interact with the genes. Architecturally, we start with the one-hot encoding of the DNA sequence concatenated with the corresponding attack-seq signal. This then passes through a series of convolution layers, which finally predict the binding affinity of various transcription factors. If you see here, we segregate the forward and the reverse orientation of CTCF and we predict so that we can also capture these orientations, which are important for the stability of TADs. To train this model, we divide the training and the testing data based on chromosomes and cell lines. So we take 3 different cell lines for training, out of which 2 are healthy cell lines and 1 is a cancerous cell line. And then we use a fourth held out cell line for testing on the unseen chromosomes 15 to 17. We show that the stage 1 of the model performs well on CTCF, YY1, and SP1 for these held out chromosomes of the new cell type. But does this mean that the model really captures the biology that we wanted to capture? To show this, we look at the deep lift attribution plots for the first layer of the CNN. And we indeed observe that the model pays more attribution to relatively open regions, while at the same time learning a motif that is very similar to the CTCF motif. And we can observe similar patterns for YY1 and SP1 as well. Now coming to stage 2. The goal of stage 2 is to predict cell type specific interactions using the features learned in stage 1, and again using the raw DNA sequence and chromatin accessibility data. In this case, we first start with 400 consecutive open chromatin peaks of the cell type that we are interested in. The reason why we now look at peaks is because for the given cell type, all the active enhancers, promoters, and most of the CTCF sites will already lie in these peaks. And this also gives us the flexibility to look at large receptive fields of interactions. The reason why we only focus on 400 is that across cell types, we observe that 400 consecutive peaks can span up to 2 mega base pair of the genome, which is indeed required to capture long-range interactions. Coming back to the architecture, so for each peak in our input sequence, we look at the DNA sequence around the peak, combine it with the corresponding attack-seq signal, and pass it through the pre-trained convolution layers from stage 1. This generates the features that we need for the given input peak, and we repeat this for all the peaks that we have in our input. So now we are at this stage where we have the features that don't depend on cell types, yet relevant for 3D folding. We pass them through a series of transformer encoder layers to capture now the relation between different attack-seq peaks from our input, and finally predict the high-C interactions between them. The way we predict the high-C interactions is very similar to how it was done in DeepC. That is, we predict the interactions around the central peak from our input sequence, and we predict the interactions only for the central 200 peaks, so that we ensure that for all the interactions that we're predicting, we do have information to the left and the right of the regions of interest. The experimental setup, again, is very similar to what we saw before. So we have a healthy cell type that we use for training, and we use the held out chromosomes for a held out cancer cell type for testing. Before we look at the results, we asked ourselves, do we really need something so complex to predict these interactions? Maybe something simpler could do a very well job as well. To do this, we defined a median model where the focus would be more on the distances between the interacting elements and not too much on the biology. To do this, we take our training data, we select all the interactions that have open regions as endpoints, and filter out based on different distances. For a given distance di, we then select the interactions that are distance di away and take the median of those interactions. And we do this for all distances that we have in our training data. Then for the test data, all we need to do is look at the distance between the two elements of interest and assign the prediction based on the median value that we obtain here. We observe that the Pearson correlation in this case is quite high for both median and UniversalEPI, although median performs slightly better. However, in our use case, we are more interested in the ranks of interacting elements. So we look at Kendall's correlation. And here we can see that UniversalEPI performs similar to the median method that we introduced. We also do introduce the effect of distances in our model through relative positional encodings of transformer. However, our hypothesis is that maybe the model is not capturing it as well. So we might want to introduce distance in a different way. Yes, and to summarize, so in this work, we implemented UniversalEPI that can predict transcription factor binding sites accurately by identifying the motifs of different transcription factors. It can capture long-range regulatory interactions by using transformers to learn the context. And it can predict interactions across cell types, as we saw that there was very little drop in performance between the same cell type performance and across cell type. And this is majorly because we combine information from stage 1, which is not specific to any given cell type. Some points of discussion or limitations is that we still need to find an efficient way to incorporate distance and the biological features. We need rigorous method testing, which includes different chromosome splits, different cell type splits to ensure that there's no bias in our data. We need some extensive method benchmarking so that we can, in a fair way, compare UniversalEPI with the existing methods. And finally, we need to have some explainability in these kind of predictive models, which we currently lack in stage 2. So we are working on that. Finally, I would like to thank Simeon, Tuna, Felix, Ignacio, and Valentina for their active contributions, as well as to the remaining computational cancer genomics group at ETH for their unwavering support. Thank you.
Thanks, Aayush. We have plenty of time, so we can have questions. And while you guys are thinking about them, I can ask one.
Yeah.
So it is a bit of a challenging task to predict the whole high C matrix, with a lot of noise and everything driven by genomic distance mainly when there's nothing else. So have you considered focusing on predicting loops or predicting pixels that are significant between ataxic peaks versus not? Those are the ones that are going to be of most interest. You predicting the background likely doesn't really serve your purpose here. So have you tried that, or have you thought about it?
Indeed, for the stage 2, we only predict the interactions between the open regions. We do not predict the complete high C matrix.
I see.
So the focus was always on predicting interactions just between the open regions.
And is that in a way that you are trying to distinguish the loops or significant interactions from the others, or you are trying to predict the continuous values?
We are trying to predict the continuous values.
Yeah, that's what I was trying to refer to. That
So the whole idea is if we have, say, a promoter and multiple enhancers interacting with this promoter, we would want to rank those enhancers to understand which one is the most affecting enhancer. And so we do predict the continuous values for different open regions.
Those continuous values would still also be driven by genomic distance that you would have to correct for to still rank it.
Yeah.
Okay.
I mean, based on the findings from our project, I'm wondering whether this first step of defining the specific transcription factors is unnecessarily restrictive, right? Because if mechanistically you just need to have an active enhancer to have contact, then does it really matter what binds there?
Well, if an enhancer is active, we would still need to know where the YY1 is binding corresponding to the new promoter.
Why?
I mean, we need to identify which promoter it interacts with, right?
So what is the underlying biological hypothesis then? That you would have YY1, for example, binding at both, and it needs to be the same transcription factor.
Right.
I mean, but is it necessarily the case? I'm just not entirely sure if it's necessarily the case.
I'm not sure either. I think I'll have to inspect that, yes.
Actually, this is an interesting discussion where I can contribute. So there is a YY1 high chip data from the original paper, I think, where they show that YY1 is enriched in enhancer promoter contexts. And so we had used this to predict an expression in using these high chip interactions and open chromatin peaks in enhancers and promoters, because we also thought that TF would bind at the enhancer and the promoter establish the link. So this is a paper that we published in the Regenerating Chromatin a few years ago, and we never found that they would bind in both. Yeah? Okay. But we did find that YY1 was enriched as a feature, so that's good. But so I think the concern that BK is mentioning seems to be valid, that it's proving too restrictive. How do you do it?
So, are you suggesting that we might need more transcription factors to capture this better?
I'm just not worried about it.
Okay. Yeah. We can test it. Yes. Yeah. GC content. Yes. So I think a part of it is captured by SP1, right?
I guess just related to this discussion, have you done like an ablation study with and without the first stage? Because it's still possible that the second stage can learn other factors like SPY1, else.
So in that case, what happens is that the second stage becomes very specific to the cell type of interest, and therefore then it's not generalizable to new cell types. So with stage 1, what we're trying to do is learn features that are ubiquitous across cell types, and so that helps. I mean, I don't have the numbers, but yes, it's low.
I mean, one of the problems of the high C is the resolution. That's still very low. And in that case, in your prediction, it's very important to see if you're able to predict the boundaries and at what resolution this is happening. Otherwise, you'll be protecting completely different types. Did you look at that?
So we are using 5 KB resolution at the moment, and we believe that for most of our interactions that we are interested in, this resolution is decent.
But the experiments themselves are lower than that 5 KB.
What do you mean?
So the high C now currently that exists experiment, I think that actually the range is a bit lower than 5 KB. So this is why actually you fixed this 5 KB, but did you look at different window size in that case to see if this is?
So we haven't focused on different resolutions yet, but that is one thing that we will check. Yes, thank you.
No other questions. One quick comment about YY1. That there's a recent work from Baptijan's lab showing that YY1 depletion, although it is energy is enhancer promoter loop, its depletion really doesn't impact neither the looping nor the transcription. At least acute depletion, a day or so. So there's kind of like a debate building around that right now. Just so you know.
But I think it's still interesting. We can check it out.
Yeah.
Thank you.